Very good morning, students. Regarding diversity of living world, today we are going to discuss Archaebacteria, Mycoplasma, and Actinomycetes. So, what is Archaebacteria? The name Archaebacteria from arche, which means ancient. It is a group of microorganisms considered to be an ancient form of life. Archaebacteria are primitive prokaryotes and are adapted to thrive in extreme environment like hot springs, high salinity, low pH, and so on. The Archaebacteria evolved separately from the bacteria and blue-green algae. Carl Woese and co-workers in the year 1990 introduced three domains of life: bacteria, archaea, eukarya. So these are all the three domains of life introduced by Carl Woese and co-workers. So what are the important characters of Archaebacteria? It is a unicellular primitive prokaryotes. Archaebacteria made up of only one cell, unicellular primitive prokaryotes. There is no well-developed nucleus and membrane-bound organelles. They are adapted to thrive in extreme environment like hot springs, high salinity, low pH, and so on. So the bacteria can live in extreme environmental condition. This is only said to be Archaebacteria. The unique feature of this group is the presence of lipid like glycerol and isopropyl ethers in their cell membrane. So ether, it's nothing but linked lipids. So this is very important characteristic feature of Archaebacteria. Due to the unique chemical composition, the cell membrane show resistance against cell wall, antibiotics, and lytic agents. Archaebacteria have very rigid cell wall. Archaebacteria cell wall made up of pseudopeptidoglycan glycoprotein or S layer. Archaea possess genes and several metabolic pathways that are more closely related to those of eukaryotes, notably for the enzymes involved in transcription and translation. So these are all the different steps involved in protein synthesis, transcription, and translation. The mode of nutrition may be autotrophs or heterotrophs. Autotrophs, mostly chemoautotrophs, can prepare their own food material by using chemical energy is said to be chemoautotrophs, or heterotrophs obtain the nourishment from other source of organism. That is said to be heterotrophs. Next, reproduction. Asexual method of reproduction take place in Archaebacteria. Example: Methanobacterium, Halobacterium, Thermoplasma. So the unique feature of this group is the presence of lipids like glycerol and isopropyl ethers in their cell membrane. Due to the unique chemical composition, the cell membrane show resistance against cell wall, antibiotics, and lytic agents. So far we discussed Archaebacteria. Next, Mycoplasma or molecules. What is Mycoplasma? The Mycoplasma are very small, 0.1 to 0.5 micrometer, pleomorphic gram-negative microorganism. So gram-negative cannot retain the stain, gram-negative microorganism. They are first isolated by Knockard and co-workers in the year 1898 from pleural fluid of cattle affected with bovine pleuropneumonia. Phytoplasma, earlier known as Mycoplasma-like organism, or MLO. They lack cell wall and appear like fried egg in culture. The DNA contains lower guanine and cytosine content than true bacteria. They cause disease in animals and also plants. In plant, Mycoplasma cause little leaf in brinjal, witch's broom in legumes, phyllodes in cloves, spike disease in sandal. So these are all some of the important plant diseases caused by Mycoplasma. Mycoplasma cause pleuropneumonia, lung disease of cattle and sheep, characterized by inflammation of the lungs, is caused by Mycoplasma mycoides. Next, Actinomycetes. Actinomycetes are nothing but actinobacteria. Actinomycetes derive from two words, actus, mycus. Actus means ray, mycus means fungus. Actinomycetes are ray fungi. So actinomycetes are also called ray fungi due to their mycelia-like growth. It is a fungus like bacteria with cylindrical cells which are usually united to form filaments resembling the mycelium of the true fungus. Actinomycetes is a gram-positive mycelial bacteria known to produce a variety of industrially and medically relevant compounds, that is antibiotics, chemotherapeutics, fungicides, herbicides, and immunosuppressants. Actinomycetes are anaerobic, can survive in the absence of oxygen, are facultative anaerobe. They do not produce an aerial mycelium. Most of the actinomycetes are saprophytes, but some are parasites. The DNA contains high guanine and cytosine content. So DNA made up of nucleotide which contains nitrogenous bases, purine and pyrimidine. Adenine, guanine are purine, thymine, cytosine are pyrimidine. Adenine always pair with thymine, guanine always pair with cytosine. So here in actinomycetes, the DNA contains high guanine and cytosine content. Example, Streptomyces. Then Actinomycetes frankia is a symbiotic actinobacterium which produces root nodules and fixes nitrogen in non-leguminous plants such as ulmus and casuarina. They produce multicellular sporangium. Actinomyces bovis grows in oral cavities and cause lumpy jaw. Streptomyces is a mycelial forming actinobacteria which lives in soil. They impart earthy odor to soil after rain, which is due to the presence of geosmin, a volatile organic compound. Streptomyces only impart earthy odor to soil after rain, which is due to the presence of geosmin, that is a volatile organic compound. Some important antibiotics, namely streptomycin, chloramphenicol, and tetracycline, are produced from this genus. So these are all the important characteristic feature of Actinomycetes, that is actinobacteria. So far we discussed Archea bacteria, Mycoplasma, and Actinomycetes. That's all about today's class. Thank you.